Thank you very much for joining this webinar co-organized by the Department of CIMUS of University of San Diego de Compostela and Sino Biological Europe. And also thank you for interest in our CRO service platform at Sino Biological. My name is Wang Chen, so I'm the host of the event. I'm also the key account manager at Sino Biological, responsible for clients in Spain, in Portugal, and in France. Before starting the presentation, I would like to introduce the director of the European subsidiary of Sino Biological, Dr. Hu. He would like to give a few words. Dr. Hu, please.
Yeah, okay, yeah, thanks, Ron. Hi, good morning, everybody. My name is Bin Hu, and I'm responsible for the Europe team of Sino Biological. And thanks for your interest here. And in this webinar, Dr. Zhang will give a detailed introduction of our company, and we certainly welcome all questions raised here, and welcome everybody contact us whenever you have a demand for our recombinant proteins, antibodies, or the related customized services here. Okay, thanks. Yeah, give back to Ron.
Thank you, Dr. Hu. So maybe we should just start with the presentation. And now I will give voice to today's speaker, Dr. Zhang Lili. Dr. Zhang, please.
Thank you. Thank you, Ron. First, I would like to share my screen. Could you see my screen, Ron?
Yes.
Okay, good. Okay, I will start now. Thank you for joining us today for this webinar. My name is Lilin Zhang. I'm currently working as a technical specialist in Sino Biological Europe. So first, I would like to take this opportunity to thank CIMUS for organizing this amazing event for us. So today, I will give you an extensive introduction of our CRO services for recombinant protein expression and antibody development. So I hope you will enjoy this event and find something interesting for you. So here is a very quick overview of our company. Sino Biological was founded in 2007, and our headquarters is located in Beijing, China, where all the manufacturing facilities are. So we also have branches in Shanghai, Guangzhou, and other cities in China. Sino has established 2 subsidiaries in the United States and Europe, respectively. So as the only official daughter company in Europe, we sit in Frankfurt with our local sales and service team, as well as our protein inventory. So our company is actually growing pretty fastly. We were recently rewarded the IPO kind of equivalent NASDAQ in the United States. And we have more than 400 employees all over the world now. 30% of us hold advanced degrees, either master degree or PhD degree. We have large lab and office spaces in Beijing. Our core business includes catalog products and customized services. So in terms of the catalog products, we can provide recombinant proteins, antibodies, genes, as well as cell culture supporting reagents. So the customized services are going to be our main topic today. I will elaborate later. Before we are getting into the main topic, I do have to mention the catalog products that we carry. With the world's largest validated recombinant protein bank, we provide a broad range of products, including drug targets, cytokines, and their receptors, Fc receptor proteins, as well as viral proteins to support the research and development in the scientific communities. So Sino Biological have independently developed more than 1,500 drug target proteins with high purity and activity to support multiple research areas and covering multiple research animal models. So we have a full collection with more than 1,000 proteins of cytokines and their receptors. The most popular targets such as VEGF, TNF-alpha, IL-6, and so on are certainly included. So Sino Biological is the largest supplier for bioactively validated Fc receptors from different species. We also have variants for the IgG binding. So Sino Biological has a complete viral protein bank. It covers more than 500 proteins from different virus families. For instance, coronavirus, influenza virus, RSV, HIV, and so on. We have the biggest collection of SARS-CoV-2 related antigen and antigen products. So in terms of CRO services, which is our main subject of the day. So this slide briefly introduces what we can do for our clients at this point. And basically, we utilize our recombinant protein production platform. We offer the recombinant protein and antibody production services. With our antibody development platforms, we also offer antibody discovery services as well, which I will talk later in today's presentation. Along with the general introductions, I will also include a few cases showcasing the work that we did in the CRO services area to give you a very comprehensive understanding of our capabilities. So this slide basically focusing on the workflow, how we do our business. When we receive an inquiry from our client, we can offer them a standard service package for some simple products. For instance, the recombinant antibody expression is considered not a very complex case, so we can provide them a standard service package, which will be followed by a standard quote. And once the payment is issued in a form of the purchase order or other payment information is confirmed, we can move forward to carry out the project. Some update will be provided by our project managers later. So we also have some non-standard service package, which means if the project is more complex than simply just antibody expression or normal protein expression, then there will be a lot more procedures to be involved. Essentially, we have to finalize the details of the project, and we have to identify the scope of the project to be very clear on the specifics of the deliverables. So if we and the clients are both happy with the proposals of the project, then we can move forward to carry out the project. I think during the process of the project, you will be notified once the project is initiated, and each milestones will be updated. So of course, once the project is finished, there will be a shipment process along with the invoice, a certificate of analysis. Definitely, we will provide a complete project report. So let's move on to the first topic of today, which is the recombinant expression. So recombinant expression is a very useful tool, not only in the scientific research area, and also therapeutics derived from recombinant proteins or recombinant antibodies are becoming more and more important in the medical field. So comparing to the nature-sourced proteins, I think recombinant proteins have some significant advantages. For instance, unlimited resources. They can be made as animal-free with high yield and low batch variations. So in order to generate a recombinant expression, we have to synthesize a target gene and clone it into an expression vector, then transfect the vector to the host cells. I think the vector will hijack the cellular machinery of the host cells to generate the target protein. So once the target protein is expressed in the host cells, we can purify the proteins using different chromatography approaches. So in Sino Biological, I think we have 4 platforms for the recombinant expression. We have I think the first one is HEC293 and the true cell lines with the mammalian cell expression system. The protein expressed can have correct fold more closely to nature post-translational modifications. They are very good at producing soluble proteins, but usually they need a longer culture time and relatively more costly compared to the prokaryotic system. I think the insect system is also useful too for the recombinant protein expression. They can be cultured to very high cell density, and they are very good for expressing like intracellular proteins and membrane proteins. But the insect cell has a very unique profile. The proteins they produced often have short glycans. So, but this could sometimes serve as advantage if you work in the field of protein crystallography and try to obtain the crystal structure of a protein, because sometimes the heavy glycosylation might influence the protein molecules packing into a crystal sometime. So we also have the prokaryotic expression system, E. coli expression system. So it can produce protein at a very rapid scale and with low cost. But these proteins produced by E. coli usually doesn't have any post-translational modifications. And sometimes if the protein are expressed in quick speed and a massive amount in the E. coli cells, they are proven to form the including bodies, so which will add some difficulties for further purification steps. For the yeast expression system, it has a similar feature as the E. coli expression system. For instance, low cost can express the recombinant protein in a very rapid manner, easy to scale up, but the proteins expressed by yeast usually with high mannose contained glycans. So the host cell selection is very important. I would consider it as a very first step in order to obtain successful recombinant protein expression. So once we receive an inquiry, we will perform a sequence analysis of the protein and try to identify these parameters of interest. For instance, the molecular weight, the PI transmembrane domain, post-translational modifications, cysteine content profile, instability, and so on, which might influence the construct as well as the downstream process associated with the expression and the purification of the target proteins, and also what kind of quality control methods and standards we are going to pursue. So after that, we are going to run a pilot to assess the feasibility to see the protein is feasible. If everything works well, I think we can scale up the expression and the deliver of the final products to our clients. So here is a pie chart showing that the whole species of the target proteins of interest that we made in the year 2019 and 2020. As you can see from the pie chart, human proteins are the major share. Of course, the viral proteins are also were also the star of the year. And we also express proteins from other species as well. So we also have extensive experience in producing proteins from upper respiratory virus strains. These proteins have been formulated into a high throughput chip-based assay platform. So if you are interested in this chip-based assay, you can look up our Sino Immune chip, and hopefully it will help you as well. For the affinity tags, they are important for either protein purification as well as enhancing the protein solubilities. So here list most of the common used tags in our company. All the tags have different size and features, so they are suitable for different systems. For instance, the histidine tag is more universal with low immunogenicity. It can enhance the protein solubility at some degree. It is also suitable for the denatured purification. So the flag tag is also available. It is suitable for the insect or mammalian cell expression systems. It has low immunogenicity with a low impact of the protein function. It also generates authentic N-termini after EK enzyme digestion. But we do not use it for the purification quite often because it is relatively costly. So GST tag is a protein tag, which is suitable for insect and E. coli. It has quite good performance for enhancing the solubility. But sometimes the GST tag proven to be degradation, so it might be a little bit risky to use this tag. So MPP is a tag suitable for insect and E. coli, and also a protein tag. It has a very good performance regarding to enhance the solubility, but it is not a purification tag. FC tag is also purification tag, and infused protein can be purified using protein A chromatography. It is good for insect and mammalian cell systems. It is also a good tag to enhance protein solubility, but due to the intrinsic disulfide bond profile with FC tag, sometimes the fused protein might be proven to aggregation. So please do pay attention if you would like to select a protein tag. Due to the space hindrance, sometimes the protein tag might mask the active site of your target protein. So I would like to consider introduce the cleavage site between the protein tags and your target protein to remove the tags in the purification steps. And now I would like to show you some cases during our work. I think a successful recombinant protein can be made using different approaches combined essentially. So in this case, it actually shows the generation of a tag-free dimeric protein using the HEC293 expression system. So this case this showcases the purification step that we undertook in order to obtain the protein with the 99% dimer and to maintain its biological activity. So the first purification step we did is to use the ion exchange column, and the fractions including the target protein were collected and further loaded into a gel filtration column. After 2 steps of gel filtration purification, we already can obtain the target protein with very high purity, and the dimer content is about 99% analyzed by the SEC. Here, in terms of the biological activity, it is validated by the cell proliferation activity. So this case tells us that sometimes a tag is not necessarily for obtaining a target protein with high purity. The second case, I would like to show how important the correct construct is going to impact the protein expression. So this one is the extracellular domain of a single-pass membrane protein that we made in the insect cell expression system. So in the first construct, we tried to secrete this protein into the cell culture supernatant, but there was no secretion at all. So then one went back and performed the hydrophobicity analysis and identified a highly hydrophobic region at the N-terminus of the sequence, so which might be the causing reason for no secretion of the protein into the supernatant. Then after discussion with our client, we agreed that this region is not relevant for the desired activity. So we removed this section with the revised construct. Here you can see from the SDS page, it showed a clear good expression. Only after 1 round of nickel column purification, we already can get the protein with high purity. And so the constructs are very important. The certain regions of the protein might need to be removed for good expression at some case. Here is the third case that we expressed a single domain of the membrane protein. And during the first extraction round, we used a certain combination of buffer and detergent, which failed to extract the protein. So then we went back, look at the alternative approaches in literature, and came up with the second round of combinations of the buffer and the detergents. This combination did successfully extracted the protein into the solution, and after a gel filtration step, we already have a very good single pass membrane domain purified, and it's stored in a buffer that contains both PBS and DDM, which is another detergent to stabilize the protein. I think this case shows that the detergent selection is very important for successful extraction of the membrane protein. The fourth case is that we try to express the nuclear capsid protein from SARS-CoV-2. Initially, we succeeded in producing this protein using the insect cell expression system. But due to some reason, the proteins existed in the mixture of oligomer, monomer, and some aggregates. So then we switched to the E. coli expression system. during the trial with the first strain, we do see the expression of the target protein, but the protein is degraded easily. So then we switched to the second strain of the E. coli. With this strain, we do see a very stable expression of the target protein. After a certain round of purification, we already get the protein with the purity more than 95%. So according to the SEC assay, the protein is indeed in a correct oligomerization state. So this case indicates us that the expression of host also plays an important role in correct recombinant protein productions. The second part is the recombinant antibody expression. So antibodies have different shapes and formats. In humans, the most abundant type is IgG, I think, followed by IgM and IgA. We also have limited amount of IgD and IgE. IgG also has some different subtypes. We have experience expressing all of these 4 types of IgG as well as IgM. We have quite a limited experience in expressing IgA. So, and also antibodies can be fragmented into the 5 SCFA and another unique class of antibody called nanobody. So we also can fuse the fragments of proteins into the Fc part of different antibody subtypes, forming the protein fusion antibodies, which can extend their half-life. There also can be chimeric antibodies or bispecific antibodies. Fortunately, we do have quite some experience in producing most of these formats. For the expression host, we use HEC293 or true cell lines. For the normal IgG expression, we just clone the light chain and the heavy chain into 2 vectors and co-transfect them into the host cells. The host cells will help us to generate the antibodies. For the tags, we normally use the histidine tag or FC tag for the affinity purification. Here are some cases showing that we can produce the full-length antibodies from different formats at a very good purity and low endotoxin levels, mainly in the monomeric conformation analyzed by the HPLC assay. Here also show some cases of antibody fusion proteins. So we can express nanobodies fused with histidine tag, fused with FC tag, also the SCFV fused with histidine tag or FC tag. We can also produce fiber in a histidine tag fused format, or we can generate a fiber by simply digested of the full-length antibody using some enzymes. So we also do have some experience in producing bispecific antibodies in different formats. So, but we do prefer the client can provide us with the sequence of the antibodies. For those ones with bispecific antibodies, we really like the sequence provided by our client. One of the featured services that will provide is to use a FUT8 double knockout HEC293 or true cell lines to produce a fucosylated antibodies, which means the fucosylated residues on the antibody glycan chain will be removed using these cell lines. And these fucosylated antibodies are very good for the ADCC activity enhancement. Here are some cases, 2 antibodies expressed either in the FUT8 double knockout HEC293 cell line or the true cell line. And basically, you can see a really difference in molecular weight or monomeric dispersion of the antibodies by SEC analysis alone. But when these antibodies are examined by the mass spectrometry, we can clearly see molecular weight shift with about 292 kilodalton on the glycan chain. So if the ADCC performance is something you are interested in, these cell lines are very good tools to generate the ADCC enhanced antibody. And another feature service for recombinant protein is to produce IgM antibodies using HEC293 system at this point. As you know, they are huge. They can be pentamer or hexamer depending on whether they have G chain or not. We have 2 methods to produce pentameric IgM antibodies, so which requires the G-chain. Both methods will able to yield antibody at correct molecular weight and high purity analyzed by the SDS page. So the expression of the hexameric IgM is quite similar to the expression of IgG antibodies as well. I think this is relatively easier compared to the expression of the pentameric IgM. So lead time, I think this might be the point that our clients are interested in. Normally, we start from gene synthesis to quality control to the final product delivery. This is basically the timeline for the expression of protein in different systems. Recombinant protein production using the E. coli system will usually takes about 6 to 8 weeks. Insect cell expression system takes a little bit longer because it has to undergo a virus package process. For antibody or protein expression in mammalian cells, it also takes about 6 to 8 weeks to complete. But please note that the proteins can be varied in different size. So certain for the genes, if the gene is very large, for instance, larger than 2000 BP, it will definitely take a little bit longer time to synthesize the gene. If it is smaller than 2000 BP, usually it will take 1 or 2 weeks to complete. We also have high throughput protein and antibody expression platform. We have high throughput primer design and synthesis platform. So we can generate a primer library and subsequently generate the vector library to express proteins or antibodies. Actually, this platform was initially established for the high throughput antibody expression. Then we expanded the capacity to the high throughput protein expression. For instance, we have a lot of mutations in the RBD domain of the SARS-CoV-2 spike protein. Then we can generate a lot of mutations using this platform. Basically, the difference of the platform between our company and other companies is that we use a shake flask to perform the high throughput expression. The volumes can be ranged from 20 to 400 milliliter, and we can produce about like 100 to 200 proteins or antibodies every week. We have a very good antibody successful rate and a faster turnaround time. If it is a very well established library, I think we can produce the final product in about 2 to 4 weeks. We do use this platform to produce several viral proteins as well. So one example is that we have been produced an antibody library from B cells for a client from a COVID-19 patient. First, we expressed about like 600 antibodies for that group, and 40 of them show a very good neutralization activity. So we scaled them up to like 10 to 300 milligrams. These antibodies were subsequently used for the animal or structural studies. Here I quoted the paper if you are interested, and you can check this literature. Again, the lead time. High throughput is a little bit different. For high throughput antibody expression at this point, with the preferred format including human IgG1, IgG4, mouse IgG2A, IgG2B, combined standard quality control, including to measure the protein concentration using SDS page for measuring the protein purity. The lead time usually takes about 3 weeks. We can also add some optional analysis listed here, but that will complicate the project a little bit more. It will have some impact on the lead time as well. So if that happens, we can communicate and re-establish the workload and re-establish the lead time as well. But please note that the lead time is reference for the antibody production. For recombinant protein expression, we would have to do some small-scale trial to prove the concept. If a protein or a few proteins can be expressed using this protocol, then we can establish the large-scale expression and purification of the target proteins. So at that time, this timeline is also referable as well. The third part of our service is antibody development. So we have a very comprehensive animal immunization platform, antibody generation and screening platform, as well as these assay platforms to identify the most suitable leads for us. Our experience reached not only in the canonical monoclonal antibody development using the hybridoma, but also we have established the phage display and single B-cell sorting method based on the monoclonal antibody development platforms. So we also have experience in developing antibodies against specific post-translational modifications, for instance, the phosphorylation. And we also have the experience in generation and anti-idle type antibodies for different purposes, for instance, ADA assessment. So as I mentioned earlier, we can use different format of antigen to generate antibodies. And we have used the antigens in the format of like a peptide. We routinely use recombinant protein as immunogen, but we also have experience using the DNA encapsulated in the lipid vesicle, as well as using the whole cell as antigens. But the DNA and the whole cell immunization is a little bit trickier. So especially for the whole cell, the cell will create a significant amount of background. So once the animal is immunized in the hybridoma, it's going to be a furin screening, limited dilution, subcloning expansion, all the way to the antibody secreted. So our aim is to create a hybridoma, stabilize the hybridoma, and finally secrete the antibodies. So for the phage display B-cell sorting, there's going to be a library construction process involved in both techniques. Eventually, there will be also several rounds planning for the phage display method, and for the sorting process, it's actually a little bit easier. The binders are sorted based on their affinity during the flow cytometry sorting process. Eventually, for these two techniques, we can deliver our clients with antibody sequence or express antibodies using the recombinant antibody expression platforms. Of course, we can do polyclonal antibody generation as well. So this is relatively straightforward, immunizing the animal. And once the titer reaches the desired value, we will sacrifice the animal and purify the polyclonal using either conventional chromatography technology, or if the immunogen is relatively easy to handle, then we can do an affinity purification as well. So the antibodies that we generate are very excellent for applications such as Western blot, HC, ELISA, paired ELISA, and some of their antibodies are proven to be able to use in the cellular function assays and FACS, so as well as to serve as affinity purification reagents. So a variety of antibodies can be generated using our platforms. So generally, it will take a little bit longer to generate antibodies. The immunogen preparation will cost some time, and the animals will cost some time to mount a sufficient immune response. So after which they can be switched to process into the antibody selection process. And really it takes about 6 to 6 months to complete a month of rapid monoclonal antibody generation service. But we do have a speedy immunization scheme, which combines our unique immunization strategy as well as adjuvants, so that we can expedite this process and finish a project in about like 3 to 4 months time. We do understand that the time is very sensitive in any research and development efforts. So we also try our best to work as quickly as possible. Here is a case that we performed for a client to develop an antibody using a single B-cell sorting technique. After sorting of the B-cell, we have identified 380 antigen-specific B-cells, and we then generated about like 142 sequence pairs, which yields 46 clones, and among which 8 are better than control antibodies. They are commercially available. I think for immunization all the way to the antibody characterization, it took us about like 26 days. So the B-cell sorting platform certainly an expedient method to generate the customized antibodies. So now I would like to give a brief introduction to our CRO service for the virology research. Unfortunately, the new emerging SARS-CoV-2 and its variants greatly changed our work and lifestyle. So Sino Biological is one of the frontiers in rapid response against the world health crisis. So in order to faster respond to the current pandemic or future pandemics, research reagents and tools, I think, are very critical at this regard. We have developed a comprehensive virus research reagent bank that contains a lot of reagents from more than 50 currently known virus strains. So we can generate recombinant viral proteins at a very fast fashion. We have included influenza proteins in about 12 days. It only took us 6 days to generate the SARS-CoV-2 RBD of the Omicron variant. And we also have a comprehensive collection of the virus antigens. Here you can see again the Sino-immune chip, which contains the many upper respiratory virus antigens. And we also have a collection of influenza virus vaccine strains from the year 2015 all the way to 2021. And also the SARS-CoV-2 spike mutants are one of our feature library as well. So we have now over 180 RBD or spike protein mutants in this collection. And we also have antibodies with hypersensitivity against these viral proteins as well. Last but not least, we do have a platform to generate high quality antibodies against SARS-CoV-2 and other viruses. So our antibodies can be highly specific against the SARS-CoV-2, but not react with other viruses. They can be highly sensitive with the sensitivity at like 42.5 picomolar per milliliter against the entire M protein. We also can generate neutralization antibodies, and these can be used as control to assess the antibody invasion phenomenon of the mutants. We also have a collection of those clinical broader spectrum entire N antibodies. They can recognize the nuclear capsid protein from different variants at equivalent specificity and affinity. So these antibodies are very useful to formulate them to the assay case to identify the nuclear capsid proteins from SARS-CoV-2. Certainly, we also have the antibodies against the different targets for the Omicron variant. So I think this is all for today. Here is the contact information of our Europe site. You are more than welcome to contact us and visit our website. Thank you.
Thank you very much, Dr. Zhang, for this great presentation. So if you have any questions, you can just ask your question in the Q&A chat. Or if you would like to, you can raise your hand. I can give the voice so that you can ask your question directly. But I already see there are a lot of questions in the question and answer chat. But before that, I will go to the question that I asked in the normal regular chat before we forget it. It is from Jing Gong, and he has a question about if you have an extensive collection of recombinant virus proteins, how do you choose the most suitable expression host for these proteins? Dr. Zhang.
Thanks, Ron. I think viral proteins are quite unique from the point of evolution. Viruses are not close to either prokaryotic or eukaryotic species. So I think these proteins sometimes are quite complicated. For instance, the spike protein of SARS-CoV-2, it is a trimeric format with heavy glycosylation. So we do see similar complex proteins from other viruses. For instance, from HIV or influenza virus. For these proteins, due to their complexity and high order of structure, we would like to use the eukaryotic expression system for the recombinant expression. But for some other virus proteins, for instance, the non-structural proteins, we also can use the prokaryotic expression system. For instance, the E. coli expression system. As I mentioned in the slide, we try to produce the nuclear capsid protein of SARS-CoV-2 using the insect cell. Unfortunately, it didn't work very well. Then finally, we switched to the prokaryotic expression system. With the E. coli expression system, we successfully prepared this protein.
Right, thank you. Now we can move to the question and answer chat. Maybe I invite Dr. Zhang to read the question and to answer them, if you prefer.
So here I see some questions. The first one is from Maria. Do you have experience in the expression of recombinant proteins in E. coli without antibiotic for the selection? I think at this point, we might be never tried with this, because sometimes without antibiotic, it is easy to get contaminated E. coli strains. It will lead to no expression of the target protein. But to be double checked, I will ask our scientist from our headquarters to reply you in email.
Yes.
The second one, I think, with regards to protein recombination, how do you overcome difficulties expressing some mammalian proteins, accumulation of proteins as incluring bodies, protease contamination from host proteins, leading to degradation of expressed protein, and also antitoxin accumulation. So, I mean, for incluring bodies, we try to moderate the temperature and try to control the low expression speed. If it is really formed as incluring body, we might do some refolding at the later purification step. For the protease contamination from host proteins lead to degradation of expressed protein. I mean, we can add some inhibitors for the protease to decrease the digestion of the target protein. So also the endotoxin accumulation. I mean, we can use some affinity purification to control the endotoxin level during the purification step.
Yeah, hello. May I add up some comment words to this question? Can I? Just about the endotoxin content. Certainly, purification afterwards, that's one step. And also, we may use just endotoxin low-level materials for the whole expression and the purification process. That's also one critical method. And this really requires that originally the expression level of this protein should be very high. Otherwise, the final yield will be very low. Yeah, that's our answer. Yeah, thanks.
We can switch to another question from Louise Blanchet. So has two questions. And the first one is, what is your capacity in terms of recombinant proteins or antibody production? And in what format are the recombinant proteins, antibodies, reagents supplied? And Dr. Zhang?
I think in terms of our capacity, we can provide recombinant proteins or antibodies in a quantity from milligrams to grams, depending on how much our clients needs. We have small scale expression such as like shake flasks, small volume bioreactors, but we also have large bioreactors with large capacity like 1,000 liters. And concerning the format that we can provide of the recombinant protein, basically they can be supplied in frozen liquid format or lyophilized powder. For the logistic reason, the lyophilized powder is much easier to be transferred, but unfortunately not all the proteins or antibodies can survive the lyophilization procedure, I think. It is a degradation process. So sometimes it will compromise the structure of the protein and cause some aggregation when the protein powder is redissolved in the buffer. So as you know, some additives can be used as protectant. But what we do is that we try to a small scale to see whether this protein is stable or safe to be processed in a lyophilization method. If it is very stable, then we can go further for the lyophilized procedure. If it is not, then we can ship them in a suitable buffer that retains the stability. Logistically, I think a lyophilized powder is the easy way shipping, but we do our best to provide the best protein quantity to our clients.
Thank you. I have another question from Jean. He asked if it's feasible to transfer hybridoma cells to Europe.
Basically, it is feasible to transfer the hybridoma to the Europe. Generally, I think the hybridoma cells will be sent with dry ice in an independent package. I think they cannot be shipped together with like recombinant proteins, antibodies, or other reagents. Of course, shipping of the hybridoma cells with dry ice will add a certain amount of the bill. Usually, like we will allocate the hybridoma cells for shipping. For instance, one batch of the hybridoma cells will be allocated into 2 tubes, each tube with like more than 10 power to 6 cells. Certainly, there can be many tubes in each package, which depends on the requirement of our clients. So according to our knowledge, so far the hybridoma cells can be sent to all the countries in the Europe that we have approached with our clients for this kind of projects.
Okay, thank you. And so far, I think we are done with all the questions we have. If Dr. Hu or Zhang, do you have anything to add? If not, I think that's it. So, well, first, thank you very much again for joining us. And I hope this presentation helped you to understand a bit more about what we do at Sino Biological and what we can offer to support your research activities. And again, if you have requests or questions, please just contact us with the information provided earlier. And then yes, thank you very much. Thank you very much, everyone, for joining this webinar and for your time. And so maybe I wish you have a nice lunch at this time. And see you next time. Have a nice afternoon.
Thank you.
Okay, thanks a lot. Thanks a lot, guys. Yeah, thank you. Thanks, Dr. Zhang. Yeah, bye-bye.
Thank you.
Bye.
Yeah.
Bye-bye.